let's talk about some analysis of some microbiology data. So very commonly when you're working with yeast or microbes, often you collect data on what would be agar or LB or microbiology plates. And oftentimes you want to capture that data and present that data in a nice figure. So here I've got 3 images of data from an experiment in yeast. And what you're essentially looking at are these colonies of yeast, and this is what's called the serial dilution, which measures the growth of yeast. And you can use this to compare different strains, how they grow with respect to one another. Now, I would just point out something that I've been pointing out for a while now with the data is your data is never going to be better than the quality of your images to start. So a really important thing is to try to get really good images on the first go. You don't want to be making figures of images that already are not superb quality images. And this is actually an example with respect to these images. These images of this data are not ideal. There's glare, so that means there was a light source, okay? There's sort of an imperfect background, so the plates are sitting on top of whatever this material is here, and this material is coming through here. All these things can confuse the viewer and make the data harder to interpret. So if you're in a scenario where you're taking actual images of data, one of the biggest problems can come from you acquiring those images and the quality at which you're acquiring the images. Okay, so you need to, when you're taking pictures, you need to take the picture and you need to look at the image and you need to say, is there anything wrong with this image that I've just collected? And if somebody was doing this properly, they would note this image is actually pretty bad because there's glare, there's this background issue, there looks like, here's the cell phone, you can see the cell phone reflected back in these images. So again, these images are not good quality, okay? And this is one reason why I show them, because you're never going to produce a figure that's better than sort of the images that you have to start with. So your first goal should be trying to get better images than things like this. And you should really look at your images with a critical eye and try multiple times over and over and over to change the settings such that you can acquire more accurate images. But needless to say, I think these images were probably taken quickly, and I just want to show you how you might assemble these in sort of a microbiology data type figure. So again, if I was making a figure for what in theory might be a paper, I'm always going to start with a size of paper, a sheet that's the size of the paper, 8.5 by 11 and 600 dpi, so that I know that the resolution is going to be high. And then I'm just going to import now these pictures into this page that I've created. So I can grab this, and I can move it in, and I can grab this, and I can move it in, and I can grab this, and I can move it in. And now I'm going to start assembling the data from this image. So the first thing I'm already noticing that the images are not taken at the same size. So this one looks smaller than these, when in reality, all these plates are exactly the same size, okay? So if I was to actually present the data like this is sort of misleading because this plate is not actually smaller than these other plates. So the first thing that needs to happen is there needs to be a scaling resize such that all the data are consistently presented. So one way you could do this is you could make the opacity of one layer weaker, and you could bring it over top and you could check, are these plates the same size? And this looks pretty good. These ones look pretty close to the same size. So these ones do not need to be resized, but this bottom one for sure is a different size. You can see it's much smaller. So I'm gonna try to make the plates align, and I'm gonna systematically and proportionately resize the image such that the data is consistent across images. Now they should be proportionately accurate. Now again, this is an issue with acquiring the images. You can actually set your capturing image system up in a way where it always acquires the images consistently, which is a really good thing to do to start. But if you don't have that, you can adjust the images such that they're proportionally accurate here by doing this technique. Now they're actually all now at this point too large, so I'm just going to make them smaller. So I'm going to highlight all these layers, and now I'm going to resize. They're all highlighted, so I'm going to resize them all consistently, shrinking them so that I can arrange the data on the figure. Now you'll note that there's some handwriting here on these plates on what you perceive as the bottom. That's actually the top of the plate. So what needs to happen now is I need to arrange these such that the data is actually from top down. So what I'm going to do is flip these images such that the data is accurately shown. So the way to do this is image adjustments. Oh, no, image rotation. I'm going to flip them vertical to get all the writing at the top. Okay, so now they're all consistently with writing at the top. And you can actually read the treatments here. This was 32 degrees Celsius, this was 34 degrees Celsius, and this was a 34 control. So now what I'm going to do is, just like we were sort of making the gels straight, I'm going to rotate these such that the data is straight. So this one, when it's rotated like this, now the data is sort of as horizontal, as perfectly horizontal as you can get it. I'm going to do the same thing here. These data are horizontal. Again, these rotations and flips, I'm never changing the data inside of the box. So these are all perfectly fine. And commonly, this is one way to present effective figures, is you have to figure out the order in which the person reading the paper should be looking at these blobs of data, right? And if you organize things properly, it makes the argument easier to understand, easier to interpret. Okay, so now we got to line these up. So the way I normally, again, this is not necessarily important. You get to choose how you line these up depending on how you want to make your argument. But I usually, when I present data like this, I'll go with sort of an ascending temperature. So this one's 32 degrees Celsius. This one is going to come next, so it's going to be 34 degrees Celsius. So what I'm doing is I'm lining up the spots. So let me zoom in here. I'm lining up the spots to get ready to crop. Okay, because I don't want this whole extra plate stuff. And now I'm actually seeing, as I zoom in closer, this could be arranged a little bit better. So I'm rotating it. So now I'm lining up this spot with this one, because this one corresponds to the same clone tested at different temperatures. Likewise with this, etc., etc., etc. Okay, so now these ones are roughly lined up for cropping. I'm going to line up the last gel now, or not gel, but plate. Line up the last plate now. And you can see by manipulating the layers, I have good ability to line stuff up now. So should be like this. This one is the same as this one, this one's the same as this one, this one's the same as this one, this one, this one, this one, this one, this one, this one down here. Okay, now they're ready to crop. So they're all lined up. So we want to crop out stuff that's not pertinent, okay, in the analysis. I'm not cropping out data. I'm cropping out extra stuff that's not pertinent to interpretation of the data. I think that's a clear way to say this. So select inverse. I'm just going to grab the eraser. Now I'm just going to erase not the data. I'm going to erase the stuff that's not pertinent to interpretation of the data. Extra stuff on the plate. Okay, so now I've cropped it from the top. Now I want to crop these equally from the bottom. So I'm going to line up the points. Okay. Now, you might not work with yeast data, so that you might be seeing this video, might think this is completely irrelevant to what I do. But it's not, in a sense that you might, in general, down the line, work with microbiology data, which is constructed on agar plates. You're looking at growth of some microorganism. So in general, sort of extract the main points that I'm saying here and just sort of apply this to things you might work on, despite the fact that you might not actually work on yeast. So I'm going to crop this horizontally. You might say that this blob could be important. You could argue that. In my opinion, it's probably just it was a drop that sort of scattered out. So I don't think it's actually important to the data. This is the actual spot. But just to be safe, I guess, I'll include that. On the side of caution. Okay, so now I'm going to crop these. I'm going to select the inverse. I'm going to grab the eraser. And on each layer, I'm going to erase the content of this figure that's irrelevant to interpretation of the data. Okay, now what you're going to see is I've constructed a nice set of uniformly aligned boxes. Okay, these are usually how serial dilutions are presented. And so what you're seeing is this one was the temperature at 32. This was the same clones and analyzed at 34, and this was the control of those same clones analyzed at 34, the positive control where they all grow. And I'm testing these in different conditions here to here. So we're not quite done yet. I made this point in one of the last ones. This is actually a color image, but the data is essentially black and white. And anytime you can present the data in a black and white format, you really kind of want to do that. So I'm going to make these data image mode grayscale. Oh, hang on. I didn't want to do that. Let me first merge the layers. Now I've merged them into one object. Let me see if I can do the grayscale now. A mode grayscale. Don't flatten. There we go. Okay, so now these data are all converted to black and white. You can see, again, I didn't change anything. I didn't change any of the points inside. I didn't change any of the interpretability of the data. I actually made it what I think is actually easier to interpret, easier to see the relevant data. Now here's where contrast adjustment is really useful, okay? Because you can see that the background of these plates is kind of gray. And a contrast adjustment here is going to be really helpful for us to see the data. Like you can actually see that there's yeast here and there's yeast here, but it's hard to see that because of the background is so gray. So I'm going to do a contrast adjustment that's going to assist in interpretability of this data. So let's try image adjustment levels. So here the histogram looks different. What you're going to see is as I move this, the blacks get blacker. And this looks much better, okay? So you can just see this is a case where the contrast adjustment is actually contributing a lot to the data interpretability. This image is much easier to interpret than this image, okay? So this is, again, a case, let me repeat, a case where contrast adjustment is going to have a big effect. And I'm pushing it to the point where I'm not pushing it beyond, okay? Beyond the point where the histogram is sort of showing the data. I'm pushing it just to that edge where it looks as good as it's going to get without changing anything inside the box. And I'm going to grab the whites. Again, bring them, make it a little bit wider. Now look at this. Now look at how nice that looks. I mean, we started, again, now the images that we started with are pretty bad. Like I commented how this thing has glare. Again, you're never going to be able to produce data that looks better than the actual images from which you start. But this to this, much better. And it's much easier for me to see the data. Now, again, you can do various adjustments now outside to sort of label your lanes. So people will usually put a text box here. Let me get Arial. I always usually make it bold. And I'm gonna put the font at about the size of each lane. So something like this, I have right aligned the text, so I can move it right up Okay, and in an actual figure, these would have pertinent data telling me what this is, 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 what this is. Right now, I'm just calling it lane 1 to 6. And again, just keeping text consistent here, I'm able to label the treatment that this particular plate was, the treatment that this particular plate was, and the treatment that this particular plate was. Okay, so now this is sort of an image that was good to go. You can put this in your notebook. It's documented well. You preserve the data. And just keep in mind, you want to always save the raw data. So you want to save these raw images that you did not edit, because a reviewer always has the potential to ask for the original images that were unedited. Okay, and you always want to write in your methods the kinds of adjustments that you've done. So I would write in my methods here, the plates were cropped and the contrast was adjusted. I would write that. But this is a good sort of example treatment of how you can maximize the visual effect of black and white data with contrast adjustment.